All right, welcome back everyone to our next general biology lecture where we're going to continue our discussions about energy and metabolism and how living things get their energy, specifically looking at the processes of cellular respiration, these catabolic pathways by which energy is produced for organisms to do all sorts of things. So we'll get into the sort of general background of this here. And get through the first half of aerobic cellular respiration, and then get into the second half of it and fermentation on the next video, the next lecture. So just to recap some of those terms from before, remember we're dealing with this one major theme of life, which is that there's this constant need for input and transfer of energy in order for life to maintain its order, to sort of counteract, to work against entropy. But that energy itself can't be created or destroyed. It just has to be transferred into other places. We also talked about different types of reactions. So remember, we have those redox reactions, which are when covalent bonds are broken apart and reformed in different positions. There's usually some transfer of electrons. Some substances are losing electrons, while other substances are gaining electrons. Oxidation is when a substance is losing an electron. Reduction is when an atom is gaining an electron. So it's that transfer of electrons over the course of a reaction that's where that transfer of energy is going on. Also to remember, we talked about endergonic versus exergonic reactions. So our endergonic reactions are those ones that require energy, where like the products of the reaction have more energy because they've taken an input of energy. So they have that positive change in G, that positive change in Gibbs free energy. These are our anabolic processes, those processes that are splitting up, the processes that are adding together multiple pieces in order to form larger molecules. So those anabolic processes are endergonic, whereas the catabolic processes are more exergonic, which is saying that they release energy and they have a negative change in free energy. So like you have what's going into the reaction has lost some amount of energy to what is outside of the reaction. So that energy is being released in some form elsewhere, usually in the form of some electrons that have been passed to another area, some other molecule, some other atoms. So getting into talking about cellular respiration. So cellular respiration is a whole series of metabolic pathways, all of which are about extracting energy from the bonds in glucose and turning it into ATP or some similar energy storing molecule. ATP is the big one, but there's some other sort of intermediate side characters in this, like NADH and NADP, some other energy storing molecules. But glucose and storing that energy from breaking apart glucose into ATP. And so this is releasing energy, and that energy is being stored in ATP in an endergonic reaction, but the breaking down of glucose is itself an exergonic reaction. It's releasing that energy, which is then stored on ATP. And this is overall, we would say it's exergonic. But the full process does vary between different organisms. So us eukaryotes, our main form of cellular respiration has two different parts. So there's like the anaerobic steps, the steps that don't require oxygen, and aerobic without oxygen is what that means. And there are the aerobic steps, the parts of respiration that require oxygen. So overall, our aerobic cellular respiration has this equation of 1 sugar plus 6 oxygens. In the very end of a long chain of processes that we'll get into, you release CO2 and water. But there's a lot of other metabolic pathways. some of which are more strictly anaerobic. So some prokaryotes go through strictly anaerobic metabolism, have a strictly anaerobic metabolism. So we call these obligate anaerobes. They can't do any form of aerobic respiration. Then there are aerobic prokaryotes and aerobic like other eukaryotes that aren't animals, obviously plants. They all do aerobic respiration, cellular respiration, fungi, protists. All of them do some form of aerobic respiration, but we still have those anaerobic parts as well. But anyway, some of these prokaryotes are strictly anaerobic. They're obligate anaerobes. So would it be fair to say that oxygen is going to be a good thing for them? Like if you're living in a world in which you can get all your energy without oxygen, is oxygen going to be a good thing for these obligate anaerobes? Well, not really, because as we'll talk about, there are a number of reasons why that that process is like having oxygen involved when you have metabolic pathways that don't use oxygen. It'll kind of interfere in things. So, just to bring this back to sort of evolutionary history, like the earliest stages of life, one thing that's worth noting is we can tell from geologic record that oxygen was at very low levels in Earth's history. So, like, for much of the first couple billion years, there really wasn't much oxygen in the environment. So because of the temperature when Earth was formed, a lot of that oxygen was really binded down into CO2, which really used, wasn't used, so that oxygen couldn't be available as a receiver of electrons, and also binded up in all sorts of rocks and minerals. And just a sort of little side note, this oxygen has these particular sort of reactions to particular colors of light, infrared light. So there are some ways of actually looking at other places, other planets where there is oxygen. We can determine whether there are oxygen on these other planets outside of our solar system by using the infrared light that they reflect and refract. So that's just a little side note. But early on, we had life, but we didn't have a lot of oxygen. So the earliest life does show up in our fossil record around 3.8 billion years ago. But oxygen wasn't really there until like 2.7 billion years ago. So there was life that was all there, but it was all pretty anaerobic. Like earliest forms of respiration in existence would have been more anaerobic respiration, anaerobic metabolic pathways for breaking down chemical energy and using it to turning it into another form of chemical energy that could do work within the cell. So these early autotrophs were probably more chemosynthetic autotrophs that were using, they probably weren't doing as much consumption of glucose and stuff because there wasn't oxygen available to where that sort of respiration was advantageous. That was until the early photosynthesizers came along. So at a certain point, certain bacteria had developed this metabolic pathway of taking light and using it to bind together different chemicals in the form of sugars and other organic chemicals, but taking light and storing that energy within the bonds of molecules. And then what that did is it produced a lot of oxygen. So these early photosynthesizers, they had an abundance of CO2 to work with, and they had light, and the other conditions of the environment became favorable to where they could start just producing a lot of oxygen as they did photosynthesis to store their sugars, to trap that chemical energy of light. So they still were probably doing anaerobic respiration. So they were producing oxygen as a byproduct of their photosynthesis, and then they were, through their own metabolic pathways, their own forms of anaerobic respiration, were breaking that down into other usable forms of energy. But the thing is, this was kind of deadly for them. So it resulted in this massive explosion in the amount of oxygen that was available. But these were anaerobic life forms. These early prokaryotes, these cyanobacteria, were still anaerobic. Like, everything does some form of cellular respiration, but they did a version of it that didn't work so well when oxygen was around. So this resulted in a huge die-off of really the first mass extinction in the history of life on this planet. So, yeah. And we could think about this, like, why is this, why did that happen? Like, what's the problem with having too much oxygen, especially if you are an anaerobic, if you live, you have a metabolism that doesn't require oxygen, and in fact is kind of suited for an environment where there is no oxygen. We got to think back to some of those properties of oxygen itself. So it's a very, it does a lot of oxidizing of other things. It will take those electrons from other things that will interrupt these the energetic favorability of these different reactions. That all those reactions that are involved in anaerobic respiration are sort of working under the assumption that there isn't something else that will be ripping off the electrons and influencing their likelihood of happening. So in the presence of oxygen, those oxidizing, like, things will get oxidized that otherwise would not have been oxidized. And this is just sort of an optional reading if you want to learn a bit more about this and, like, the mechanisms of how oxygen is directly toxic to these anaerobes, these anaerobic microbes, and the various things they do to protect themselves from reactions with oxygen. I encourage you to take a look at this manuscript from a few years ago. It's pretty cool. I need to look at it myself, but I hear it's pretty cool. So at this point, I want to check that we have an idea of cellular respiration. That whole series, it's kind of a variable series, but like these sets of metabolic pathways for breaking down chemical energy into a more actionable form of energy like ATP, and that there are aerobic forms of metabolism and anaerobic forms of metabolism. Aerobic respiration and anaerobic respiration, with oxygen, without oxygen. So let's talk about the first of the big steps. So, like I said, this is a whole series of metabolic pathways. You often see aerobic cellular respiration as that one big equation of sugar plus oxygen, you get CO2 and water. But really, a lot of those reactions that we are talking about have a lot of steps. So the whole cellular respiration is a number of cycles, but at its core, it has to start with the breaking down of glucose. So this is what we refer to as glycolysis, just the breaking of glucose, glucolysis. So this is an anaerobic process. So when we say that even aerobic respirators, like us eukaryotes, do both anaerobic and aerobic things, like we're doing glycolysis, prokaryotes are doing glycolysis. It's happening everywhere. So its general Summary is this. So we have glucose going in. It takes a couple of ATP, and then at various steps along the way, it produces some other ATP as well as these NADH, which is another nucleotide-based energy transferring molecule. It doesn't store as much energy as ATP, you would say. But yeah, so it takes a couple, and then it produces some more energy. And at the very end of it, you have these two pyruvate molecules, which you see are each 3 carbons versus glucose, which was 6 carbons to start. So 2 3-carbon molecules, 3-carbon pyruvates. And you net a couple of NADH and a couple of ATP. So you would say that the ATP is initially there to provide the activation energy for this overall reaction. Remember, this overall reaction has a bunch of different reactions within it. And then at the end, it produces 4 plus 2 plus 2 NADH and 2 pyruvate. So we say it nets 2 ATP. But these are a little bit more, let's get a little bit more into the details of these steps, because glycolysis has a lot of detail to it. So really what's going on first is you have your glucose here on the left, and then you're going to add energy to the molecule in the form of ATP. That's going to add a phosphate onto it. And then that is going to get converted over into a different sugar. You see, that's a glucose 6-phosphate. This is a fructose 6-phosphate. It just involves some swapping of the oxygen and the OH, the hydroxyl groups. So things are getting rearranged on it. So add on that phosphate with the help of ATP. That phosphate is carrying the electrons. It's carrying the energy away from the ATP, and the electrons that come with that phosphate are providing the energy to this glucose 6-phosphate molecule, which is converted to this fructose 6-phosphate, which then, similar process, another phosphate is added on, so you get this fructose 1,6 biphosphate. The 6 and the 1,6, those are referring to which carbon in this chain that the phosphate is attached to. So this is saying there's 2 phosphates attached to those 2 places. So, yeah, so just to go back to here, what's going on is first that energy is allowing, let's stick over here. So we have fructose 1,6 biphosphate, and then that energy that is added there is going to split this into 2 3-carbon molecules that each have a phosphate on them. So they're slightly different molecules technically. But one thing that's here, so those are going to move on to the next step. One thing that's worth noting is along this whole chain, there's an enzyme for every one of these reactions. So you have hexokinase, phosphoglucose isomerase, and so and so and so. So like there's every one of these steps has an enzyme that is there to reduce the activation energy of these things. And in some case, there is still some activation energy from ATP that is required. So that's steps 1 through 5 of glycolysis. We're halfway there. Now, part 2, we see this is just a continuation down. We have, this is a part of the pathway that happens twice, because we have 2 of these glyceraldehyde 3-phosphates going into it now. Those get another phosphate group added to them from NADP, which is another energy storing molecule. It stores a phosphate on it. This is, again, a nucleotide-based phosphate, a nucleotide and phosphate sort of molecule. So it's going to donate this phosphate, donate that bit of energy to this, and then that will produce an NADH, which will then go on to be used for another reaction. So the phosphate is being donated, but trons are still remaining, or actually the energy is still actually increasing on that NADH. Sorry, I'm getting a little turnaround here. Glycolysis and cellular respiration is a lot to deal with, so don't worry don't worry if you don't catch all this on the first, second, or 100th time. So a second phosphate group is added from the NADP, converts it to NADH, which goes on to be used later on. We'll see it in later parts of cellular respiration. And then there's several more reactions that are involved to remove those two phosphate groups that are now on each of these. Their energy is stored in the form of ATP, water is released, and then what you get is some final molecule, which is this pyruvate here. So that's glycolysis, steps 1 through 10. So remember, this all is happening twice. So we're getting our 2 NADH here. We're getting 1, 2, and 3, 4 ATPs here. That gives us our overall, and then we have 2 pyruvates at the end. So that's glycolysis. It's going on within every organism that breaks down sugars. Just to get a visual sense of what this sort of looks like, we'll watch this video that'll talk about this important metabolic pathway and how it functions.
Biological organisms require energy to survive. Glycolysis is one of the pathways cells use to transform sugars like glucose into biochemical energy in the form of ATP. In the cytosol of the cell, glycolysis converts glucose into pyruvate through a series of 10 enzymatic reactions. This process produces ATP, along with other products such as NADH, that can be used later to produce even more ATP for the cell. Let's watch as these enzymes oxidize one glucose molecule into 2 pyruvate molecules. First, a kinase reaction adds a phosphate onto glucose to form glucose 6-phosphate. This is one of 2 energy consumption steps and is an irreversible reaction. Next, an isomerase reaction converts glucose 6-phosphate into fructose 6-phosphate by rearranging covalent bonds. Another kinase removes a phosphate group from ATP and gives it to fructose 6-phosphate to form fructose 1,6-bisphosphate. This is the second energy consumption step and an irreversible reaction. In the fourth step of glycolysis, a lyase reaction splits the 6-carbon fructose 1,6-bisphosphate into 2 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate is rearranged by another isomerase to form a second glyceraldehyde 3-phosphate. At this point in glycolysis, glucose has been metabolized into 2 glyceraldehyde 3-phosphates and 2 ATPs have been consumed. The next 5 steps of glycolysis are the energy-producing phase. In step 6, both glyceraldehyde 3-phosphates are oxidized to 1,3-bisphosphoglycerate by a dehydrogenase. This step produces 1 NADH for each oxidized glyceraldehyde 3-phosphate, for a total of 2 NADHs. These NADHs are later used to produce more ATP for the cell. In step 7, a kinase transfers a phosphate from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. This step is reversible even though ATP is formed. The next step involves a mutase reaction that moves the phosphate on the 3rd carbon of 3-phosphoglycerate to the 2nd carbon position to form 2-phosphoglycerate. In step 9, a lyase reaction removes water from 2-phosphoglycerate to form phosphoenol pyruvate. In the final step of glycolysis, a kinase reaction removes the phosphate group from phosphoenol pyruvate and donates it to ADP to form ATP and pyruvate. Like reactions 1 and 3, this step is irreversible. At this point, 2 pyruvate molecules, 4 ATPs, and 2 NADHs are formed for each glucose that was broken down in glycolysis. The pyruvates and NADHs could be used in aerobic respiration to produce more energy for the cell. Here, we depict glycolysis as a closed process. But in cells, substrates produced by other reactions can enter glycolysis at different points. For example, when an animal breaks down glycogen, glucose 6-phosphate is produced and can then enter the glycolysis pathway at the second step. Importantly, this means one less ATP is required for the pathway because the first ATP consuming step is skipped. Other sugars can also enter the glycolysis pathway at different points, each having a different effect on the net number of ATPs that are produced by glycolysis. These ATPs are important energy molecules required for many biochemical pathways and ultimately life itself. Glycolysis is a major contributor to the pool of ATP used in these pathways that are essential to the survival of biological organisms.
So hopefully that is helpful to see. I think it's important to emphasize a couple points from it. One is that we don't need, at this stage in our biology education, we don't need to have a very detailed understanding of we don't have to memorize all these names of all these different molecules, but we want to get the sense of the story. What are the big pieces that are being produced, the bigger players, the bigger ins and outs, but also recognizing some of these things of this isn't a closed system. We have this sort of model for how this metabolic pathway normally occurs. We have our energy consuming and our energy producing sides of glycolysis, these different stages. But it's important to remember that there's all sorts of other molecules. This is occurring within the cytosol of the cell, just within the general soupiness of the cell. And then there are other molecules in there that can get incorporated. It's not always glucose. It's very often not just glucose that is getting broken down to produce energy. So really, in reality, we have a variety of different things that we're consuming that our body is trying to break down, and they're entered into these different areas, and we've got some differences in how these pathways go. Metabolic pathways get quite complicated when you really get into the details of them. But at this point, we just want to get a general sense for the story. So at this point, we've taken that glucose with the help of a bunch of different enzymes and a little bit of activation energy. We've produced some energy without the need for any organelle or anything, without the need for oxygen. We've done this anaerobic breaking down of glucose into a couple pyruvate molecules, and we netted a couple ATP and NADH, a couple energy-containing molecules. So in your own time, just want you to sketch this out yourself. So sketch out those 10 steps of glycolysis, what you put along each of the pieces. You can leave that up to yourself, how detailed you want to get. But we want to note some of those things, okay, where's dehydration synthesis occurring? Where's hydrolysis occurring? Where are the more anabolic parts? Where are the catabolic parts? Where's energy being released? The exergonic reactions? Where are the endergonic reactions? The reactions that are requiring energy? Get a sense of, try to break it down and write it out yourself. We can talk about this a bit more as when we're in lab in person. So at the end of glycolysis, this is what we'll finish up with today. what we have is pyruvate NADH. So these will be very important in some of the next steps of getting energy out of these molecules. So glycolysis itself doesn't produce a lot of ATP. You only get a couple ATP for each sugar. 4 were made, 2 were used, so you only net 2. But then you have pyruvate, which can then be broken down even further. So that can be broken down through an anaerobic process, specifically a fermentation process, or it can be broken down in an aerobic way with oxygen. So you break down that pyruvate, it enters the next stage in our story, which is the citric acid cycle. So in the next video, we'll get into talking about fermentation and the citric acid cycle.